It's my pleasure to welcome Trygve. He's got his PhD in marine robotics, and his main research focus has been on effective information gathering strategies using autonomous underwater robotics. He's also a CEO and founder of a startup, Scraft Technologies, that works on designing software solutions for autonomous robotic solutions in the marine environment. So with no further delay, the floor is yours.
Thank you very much. First of all, thank you for inviting me to speak. Yes, my name is Trygve Fossum. I'm a researcher at the Applied Underwater Robotics Laboratory at the Norwegian University of Science and Technology. Today we're going to talk about adaptive sampling from the perspective of autonomous underwater vehicles, and we'll specifically talk about some work where we did try to volumetrically map microorganisms in the water column. So this is the layout of the talk. I'll give a brief introduction to myself and my research group, and then some introductions to some important topics for this talk, and then we'll dive into some actual real research, and then we'll quickly have a brief look at some of my current work, which is an extension of what we'll see here today. So my background is in marine cybernetics automation. I have a PhD in marine robotics. My topic was on AI systems and decision making and effective information gathering techniques using robotic platforms. Most of that work was done here in Norway, but a big part of it was also done in Porto and California. For those of you, I'm not sure how familiar you are with AUVs or autonomous underwater vehicles. That is the main platform I have been using, but there is one to the right in the picture. So it's essentially a torpedo-shaped vessel that carries a number of sensors and flies through the water column and can either do sampling in the water column itself or the seafloor. The way one traditionally operates the AUV is to pre-program the AUV to follow a certain set of waypoints, and then that is a deterministic mission that has a start and end point. So my work has been looking at ways to see if you can optimize the data gathering while the mission is ongoing on the basis of the data that you're gathering. And we'll see some examples of that today. The overarching goal of what I am doing, and there is actually a small group of people doing this all over the world, is to essentially provide cost-effective tools and techniques and processes for doing ocean-based measurements using robotic platforms. And it's, of course, an evolution of the history of ocean sensing that has come to a point where robotics is gradually taking a bigger role. But it's still a very multidisciplinary field, I would say. So when I have worked with this, I have worked with robotics, information theory, statistics, and had a lot of discussions with oceanographers and biologists because they are the ones that have the scientific questions that needs answering. So, and that is really the motivation for introducing robotics in the first place. Our research group is a part of the Norwegian University of Science and Technology, and we have close collaboration with a number of these different departments. So biology, oceanography, and even archaeology, is we are closely cooperating with them in order to tackle different scientific questions with robotics. We are gradually building out a fleet of a robotic sensing network. We have a number of underwater assets and infrastructure on the seafloor, and ships and ROVs, and we are also now extending this to dedicated satellites, which will be a part of this network. And this you see here in the image is the Ocean Lab illustration of Trondheim, the city where the university is situated in the background, and the fjord right outside that city. I have borrowed this slide from Kanar Rayan, which was an international chair at our university, and it's really, let's say, the future vision for our robotic activities. So, you know, Norway has a vast stretch of ocean that needs to be monitored and surveyed, and in order for us to do that effectively, we need to combine several different sensing assets. And Kana here has made a stack of a number of different platforms. So at the bottom you see AUVs, you have surface vehicles that work on the seafloor, not the seafloor, but the sea surface. You have aerial vehicle and satellites. And of course, these things observe the ocean at different scale, and the idea is that, of course, that you can pinpoint with the satellite where you actually need to do more detailed measurements, and then deploy each of these assets accordingly. And we are getting there, but it's of course a very it's still a way to go. But I think it's a nice vision, and one that hopefully will become realistic in a not too long time. There is, of course, a number of technical challenges related to this vision, and one of them is autonomy, and we'll discuss that one today. I will also make you aware of some of the sampling challenges that is related to doing ocean exploration. So I'm not sure how familiar you are with doing measurements in the ocean, but it's probably the one domain where it's actually very challenging to do data collection. So for one thing, the ocean is large, it's a dynamic environment. It's also a very harsh and expensive place to do operations. So this has a few consequences. Measurements are few and far apart. We stand the risk of undersampling the process that we want to study. It's hard to maintain up-to-date knowledge of the environment. And of course, it's a complex environment, not only because of we cannot communicate using high bandwidth channels, but it's also high pressure, currents, etc. So there is of course a question that arises amidst these challenges is where and when should we measure in order to effectively retrieve relevant and useful information. And this question sort of underlines how we approach scientific questions and how we design our algorithms and put them eventually into a platform and start to actually do the data gathering. I will also take some time to sort of motivate why robotics is gradually becoming more and more a part of ocean sensing practice. So let's say you have this subsurface plume. It has a spatial extent that is interesting to know. Maybe it's pollution. Maybe it is wastewater from a city. And it is of interest to know how much of the surrounding area is affected by this plume. So if you would use a traditional ship, you would do a cast with a wire outside the ship, and you would essentially make this profile. And during, let's say, 8 hours, you would have these 12 profiles. And it's evident here that you stand the risk of undersampling, and it's very hard to see based on this data where the plume actually, how the plume is actually distributed. If we try to imagine doing this with 10 AUVs, this is some of the results we could get. And of course, this is seems like it's far-fetched or maybe futuristic, but this is actually currently totally possible. And this shows that robotics can offer something that traditional ocean sensing practices don't. And that's why they're making a big, let's say, entry into this field. Okay, so we have talked about the challenges facing data collection in the ocean. We have talked about robotics. So why do we need autonomy? Well, in the last example, where you had 10 AUVs, they were essentially going straight, and you had 10 of them. Maybe you just have one, and you have to pre-program the AUV to follow a certain depth. You stand the risk, of course, of planning the mission at a depth that is not very useful, and you might miss some important data. So building autonomy that can react to changes in using its sensors is important, because you can, of course, retrieve much more relevant data. And you also will, of course, get a much better understanding of how things are. So if you would follow this red line, you would assume that there is no biomass in this patch of ocean. But if you allowed the sensor and the AUV to maybe adapt to sensor measurements, you would see that there's actually quite a lot, but it's just you happen to miss it. Autonomy, you can also use autonomy to be opportunistic. So if there's suddenly an alternative plan that allows you to follow another feature that you haven't seen, you can choose to do that. There is a very interesting paper by Yogesh at Woods Hole talking about this. Suggest that is something you look at. Another thing is that using autonomy, you essentially can track certain processes in a Lagrangian frame. So one paper here discusses that. And one example where this could be useful is, for example, in this video, which I personally shot from my friend's cabin. You see there is this tidal bore coming into the fjord, and there is a lot of interesting activity happening just in the border between the two water masses. It's of course infeasible to plan where this thing will be before you come to this place. So tracking processes like this has enormous value, and you have to use autonomy to do that. Finally, thing that is, of course, also possible is to alternate the sampling resolution. And this is useful because then you can avoid sampling redundant data and increasing sampling resolution where it's needed. And this ties back to what I said about undersampling. So just to summarize that, if you use autonomy or adaptive sampling, you can capitalize on the information that is in the field, and you can use this to get more scientific relevant data. And I will say you can also think of it as increasing the prospect of retrieving more scientific relevant data. So that was a motivation. Now let's look at how an approach like this can work. So usually you start out with some prior information. Maybe you know something about the temperature field, and maybe this temperature field is correlated with some process that you want to study. You can build, of course, into the platform before you go out, so that the AAV doesn't start on scratch, but maybe it has some idea of the like statistical normal situation in the place that you want to sample. So you start with some prior information, you start gathering data, and you can use this data to update your representation of the environment. Personally, I have been using Gaussian processes, which is a stochastic process where you can incorporate uncertainty and expected value in a spatial model. And it's shown here in figure C that you can see we have gathered some data and we have updated figure A with this new data, and we also in figure B can have a track of how certain we are that our measurements are actually close to being this value. So now that we have an updated understanding of our environment, we can go on to generate a new sensing strategy and use this to gather more data that is either to gather more information about the environment, or you can also say, I know enough about the environment to do targeted sampling. And this is the exploration, exploitation trade-off that you have. But either way, this is a cyclic process that at least allows you to refine your sensing strategy towards something that is maybe better than doing a pre-made plan deterministic. So this is a very brief way of looking at it, but it gets the major functionality. Okay, that was some introduction. Now let's talk about some work. This was research we did a few years ago. I did it with all of these people here, and it was concerned about doing volumetric mapping with AAVs and volumetric mapping of microorganisms, specifically phytoplankton. You see an illustration of the AAV that we used for this. In the front, you can see some red, green, and blue light. And the sensor we use to measure the concentration of phytoplankton is actually an optical sensor that uses scattered light to say something about the content of chlorophyll A in the water. And chlorophyll A is a proxy variable for phytoplankton. So if you have more phytoplankton, you will see that with increased chlorophyll A concentration in the water. We'll talk more about this as we go along. The whole reason why we went there in the first place was actually quite interesting, because the place where we did this study was actually a bird island, and this bird island had a very large population of puffin birds. The puffin looks like this. One problem was that the population of puffin had decreased significantly over the years, and nobody really knew why. And the puffin eats this fish, which it has in its mouth in this image. So one motivation for going there in the first place was to see, could we discover something about the consequences why these birds are not here anymore, or they're there but the population has declined significantly. On the left you see a satellite image showing you chlorophyll A concentration based on satellite measurements, and the island is circled in black, and you see there's these swirls of high concentration. And what is fascinating with these swirls and these patterns is that they have a fractal property. So if you zoom in, you will see sort of the same type of patchiness of chlorophyll A concentration as you zoom in. One of the goals that was given to me that was looking towards the consequences why the birds were going away was try to map phytoplankton, which is at the base of the marine food web, and see if we can say something about the state of the food web from the phytoplankton level. So in order to understand what was happening there, we needed to look at the local food web, and at the bottom of this food web is phytoplankton. I didn't know that much about phytoplankton personally before this, but it's actually plants that live in the water, suspended in the water, and it uses photosynthesis to grow. They are eaten by zooplankton, which is again eaten by this fish that the bird eat. Another factor in this ecosystem was that this fish lives inside areas where you have kelp, and this area was targeted for seaweed harvesting for pharmaceutical industry. So it was also a hypothesis that because we were taking out seaweed, we were essentially removing places for the fish to be, and then since there was no more fish, there was no more birds. But it could also be that there was some trouble with the concentration of phytoplankton or zooplankton. This is, of course, me speaking as an engineer. Please keep in mind this. It's much more complicated, but bear with me. So, okay, we had control on the seaweed harvesting, but we were interested in the zooplankton and the phytoplankton. So in order to understand and measure zooplankton, we had this ship with a frame, and on this frame was a camera that was essentially a microscope looking at particles in the water, and it could actually recognize and count zooplankton species. And it could do that over different depths. So we could get a sense of, okay, which types of zooplankton are situated at which depth and how concentrated are them. And the AOE task was to sort of make a map of the phytoplankton in the same area. So we would get zooplankton measurements and phytoplankton measurements in the same place, and we would try to put these two together in order to understand something about their interaction and their current state. And this is the sensor I talked about with the green, blue, and red light. So it measures chlorophyll A, which is a thing that these plants use for photosynthesis. So measuring chlorophyll, you are indirectly measuring phytoplankton. Okay, so we wanted to understand zooplankton and phytoplankton, the ecosystem structure. We also want to understand how these two things are distributed so that when we make a measurement, we know how patchy the ocean at this location is. So if we're measuring in the wrong place, we know that this might be an effect of patchiness. We would also try to come as close as a snapshot of this volume as possible, which is a challenge, of course, because the ocean is moving things about, and getting a snapshot is not really realistic. So we cannot visit all the places in that volume, so we have to prioritize. And there we have the adaptive sampling come in. So in this volume, we have to prioritize where the AUV should be situated so that we can make a snapshot map of the distribution of phytoplankton. And we want that map to have the highest accuracy where the peak distribution is. So the map should be very accurate where the concentration of phytoplankton is high. Okay, so it was complicated. It was a complicated task, but we devised a method that worked to some degree, at least. The method was like this. So we have the volume, and we start by exploring. So we go up and down one envelope along the side of this volume, and we update our estimation of the depth of the highest concentration inside of this volume. So we went along the sides and we updated for each envelope. We updated our estimate of how the peak distribution was inside of the volume. So we went along the sides of the volume. We then made a map of the peak distribution inside the volume. And then we were finished with the exploring part, and then we wanted to exploit this information. We went inside the volume along a predefined track, but we alternated the depth to follow the peak distribution. That way, we prioritized the most important parts inside of the volume so that the map had the highest accuracy where the concentration was highest. So it was a predefined track, let's say location-wise, but we alternated the depth. Looking from above, it looks like this. So we would go along the sides and update our estimate of the peak layer. And then in the next phase, we would use this to track along these depths in order to make a map of the phytoplankton. In the paper published, we had this figure, and it's not very easy to understand, but it's the best you can do. So for each envelope, you find the peak, the depth with the highest concentration, and you then assimilate this depth into a 3D surface that sort of tries to map this peak distribution. So for each envelope, you did that, and then in the next phase, you tracked along that on the inside. So the results from these looks like this. So in figure A, the first one, going along the sides, we had this image of the distribution. And after tracking on the inside, we updated this somewhat. You see that one of these contour volumes increased in size, but the first one is somewhat the same. You can see on figure C and for figure B that the AV is going along this hourglass track, but it's alternating its depth. And you see it marked with the asterisk. You can see the AUV making a depth change that allows it to stay within this area of high concentration. So we handed this results over to the biologists, and they used this to say something about the patchiness and something about the ecosystem structure together with the zooplankton data that they had. we combined a lot of measurements, but those were the main ones for this, for our part at least. So here is a short snapshot of the AUV doing its thing in around this island. Currently, there is we have not concluded on the real reason why the birds have left. The biologists are still looking at the data, so I'm afraid I cannot tell you the end of the story, but the seaweed harvesting has moved away from the island, close proximity to the island at least. Before I finish, I just want to say something about our latest work. So in the ocean, it's all about prioritizing where and when we should measure, and oftentimes it's very interesting in the interface between different water masses or between different bodies of water. So we have, in our recent paper, developed a tool and method to study feature boundaries and develop strategies to sample so that the uncertainty about these boundaries is reduced. So you're essentially tracking along the boundary. And this is a paper that is already available on preprint, but it's in revision for the Annals of Applied Statistics. So it's not directly a continuation of the work, but it's very related. So with that, I leave you with this short video of the AUV, and thank you for the attention.
Great, thank you so much. This has been, you're addressing a really important problem in sampling. And as you mentioned, it seems like it's you guys have made some progress. So before we open up the floor for questions and people can type the questions in the chat, I wanted to actually ask a couple of questions on my own. So the technique you described, doesn't it depend on the initial cube volume you're starting with? Because if your cube volume is too big, then you might not be able to cover all the samples within the cube, right? So how do you determine the cube volume, the initial volume?
Yeah. So that is a very good question. So you have to somewhat estimate the spatial initial spatial correlation of the phytoplankton. So we did some missions prior to this, and we had some scientific papers pointing to a correlation of about 1 kilometer. And in order to effectively make a good estimate of the size of the cube, you would also need to take into account the currents. So if there was very strong currents, we would have to have a very small cube. With the currents that we had, we could actually have a cube that was the size that if we made a measurement at the far end of the cube, it would still have a correlation at the point furthest from that point on the cube. I don't know if.
So question about the currents, does that potentially depend on the depth as well, right? Because the currents might move stuff faster at the top than as you go deeper. So how do you account for that?
So phytoplankton is usually concentrated in the surface because they need light. They can, in certain processes, be forced deeper down because of strong oceanic currents. But phytoplankton concentration is usually in the top 50 meters. So understanding how the current profile is from the surface down to about 50 meters is the most important when studying this type of feature. If you would have currents that would go in opposite directions, you would need to have, I think you need to have done things a bit differently, and you would need to have a different correlation function for those two different depths. So you would have, we had an isotropic correlation function, which means it doesn't depend on a certain direction. It's the same in all directions. We have accounted for stratification, but not current, the heading of the current in the current direction. So this is one thing that could be done to improve the statistical model.
I see. And there's another question I had about the, before I ask, there are lots of questions on the chat as well. But before I go there, can you talk a little bit more about the sensor design itself? You mentioned that there's a microscope which can give you information about the planktons, but also you also mentioned that there's a chlorophyll A sensor which has different LEDs. So how do both of them interact with each other? Is it that you're shining LEDs and then looking at the reflections, scattering using a microscope, or are these both different channels?
So the microscope, which is named the SILCAM, was deployed from the boat. So that was not on the AUV. We have later on we have put it on the AUV, but that is another story. So in this experiment, the microscope was on the boat, and that did profiles. So that had the microscope on a frame, and that essentially just wired, essentially like a fishing rod. It just sent the frame to the sea to a certain depth, and then take it up to the surface again, and then would profile the concentration of different species at different depths. And it uses a convolutional neural net that is trained to recognize different types of zooplankton. And that camera can only see zooplankton because the magnification is not high enough to see phytoplankton. So phytoplankton is even smaller, so that you cannot see that with that microscope. There's other microscopes that can, but this one could not. So the AUV was used to look at the phytoplankton, which was much smaller, and that uses an optical sensor called the EcoPuck. And the EcoPuck looks at chlorophyll A, and chlorophyll A is a proxy for phytoplankton, and it measures the concentration of chlorophyll A by shining a light at a certain wavelength and looking at the scattered reflection of that scattered light. And the amount of scattered light that's coming back tells you something about the amount of chlorophyll A.
Cool. Yeah. And you said that the neural network was trained to identify the zooplanktons. How did you get is it something which exists? Does the neural network exist from other work, or how did you get the training data, the labeling and other things?
So it's actually a big community doing oceanographic microscope, or microscope for looking at different types of organisms. So there are open source models that you can use, but for this one, they actually annotated the data themselves, and they did data augmentation to increase the size of the data set and stuff, but they actually annotated that themselves. I'm not sure how many pictures they had in the training set, but I guess someone spent their summer on doing that.
So we're going to go to the questions in the chat. Amy has the first question on, did you use only one robotic AUV per cube, or did you have multiple AUVs? And if so, how did you determine the path planning and the current location within the cube so that two robots don't overlap at the same time? Or they,
Yeah. Was it more?
yeah, that's basically the question.
Okay. Yeah, sure. No, we had only one AUV. But on that note, if we had two, it would be a very different case. Because if we had two, we could, of course, have some sort of dual mapping scheme where the other one would feed maybe some information to the other AAV, and then they could collaborate to map this essentially twice as fast, at least. So we only had one AAV. I know that people have done similar stuff using two AAVs at the Monterey Bay Aquarium Research Institute. They just published a paper on that, which is truly it's much better than my work, but they actually tracked this for several kilometers using two AAVs and the surface platform. So we only had one AAV, and we had to do the best we could with that.
The next question is from Alex, who was asking about how does the moving through different biomasses affect the sensor sensitivity and specificity? For example, if you have a thick school of fishes which can reflect light or other particles, how good is your sensor?
I cannot answer that fully, but for the optical sensor, it looks at the backscatter or the reflected light, and it looks at the backscatter on a very short distance. It looks just some centimeters outside the AUV, and a school of fish would not interfere with that measurement. What could interfere with that measurement is other suspended or dissolved material in the water. The thing is that this sensor, in addition to measuring chlorophyll, it's also measuring two other parameters, which is the color dissolved organic matter, and it's also measuring something which is color dissolved organic and total suspended matter. So it sort of looks at the color of the water. So if it's water or tea or coffee, if you get that analogy, and it also looks at the particle distribution or the particle. It has an estimate of the particles in the water. So when it measures the chlorophyll A, it sort of correlates and checks these two other variables so that it's giving you the correct concentration. More than that, I don't know.
Cool. So Hannah has a question about the last project you described on your last slide. How will the output of the ongoing project you just mentioned, which is on reducing feature uncertainty, be integrated into motion planning for AUVs?
So for that one, essentially there is a, let's say, challenge, still ongoing challenge in ocean sensing, where you have to essentially put the AUV as close to the boundary as you can. The trouble is that along boundaries, correlations can be very tricky. So it's hard to infer how things look around you. So reconstructing information in locations where you have not been is difficult. So what our paper here does is to sort of give you a statistical representation or a statistical measure of the uncertainty of where this boundary is located, so that in order to reduce that uncertainty, that measure or that metric is telling you, in order to reduce the uncertainty about where this feature actually is, you should put your AUV at this location. And depending on how many, how you have discretized and how many steps into the future you look, you can optimize the path of the AUV accordingly. The trouble is that it's quite computationally heavy. So if you're looking several choices and several possible choices into the future, you have an exponential growth, and you have to sort of prune out some of the path eventually, because you have so many subsequent choices and alternatives. So that is like also a challenge that is discussed in this paper, but in this work and the feature boundary work in this slide here, it's giving you something new that previously has not been done. It's giving you a way to describe the uncertainty about where these features boundaries are, which is a very useful tool if you're looking at certain types of ocean processes.
So Laurel has a question, which is on the title slide of your presentation. It looks like you're deploying the AUV by hand. Is that normal, or do you have other deployment mechanisms for doing so?
So we usually don't throw them off ships, but sometimes it's actually, if it's not too high, it's actually a safe way of deploying them, because hanging a person over the side of the boat can be not good in terms of security and safety. So sometimes we just throw them off the boat. And this deployment is in the Arctic, so the water is very cold. So usually we actually just lower them down on the side, and sometimes we even use a crane. It looks very cool when you throw them, but if you get like more than 3 meters, you can actually damage the AUV.
Nice. I had another question about the computation you're doing on the AUV itself. It's a closed loop system, right? It's figuring out what the concentration of the planktons is and then trying to figure out its path. So can you speak more broadly about what is the computational complexity of things you can run on the AUV itself? Can it run deep neural networks on it? What's allowed on this platform?
So you could compare it essentially to an aerial drone, or the computational capacity is actually quite good, I would say. So in this AUV we had the NVIDIA Jetson TX2, and that is fully capable of running quite large structures. It's of course depends on how large your network is, but for GPU type of stuff, it's actually quite okay in terms of computational availability. If we're doing things that are not GPU based, so one thing I just talked about was the looking, if you discretize your operation area into certain nodes that you can visit, you have an exponential growth of complexity in terms of waypoint choices. So in order to evaluate longer routes, you run up to a computational problem, because there are very many routes to evaluate, and if you're evaluating an objective function that has complex terms that may be requiring you to inverse large matrices or find the linear algebra is heavy, then you can of course have a problem. And the reason we use statistical models on board these platforms is that fully numerical ocean models that solves the Navier-Stokes equation, which is the equation that tells you how water particles interact, that is way too heavy. That has to run on a supercomputer similar to the ones that predict weather. The reason we use statistical models is because they are computationally tractable on board AUVs.
Cool.
That's.
So Matthew has a question on the various parameters of which you had in the Gaussian process, like the hyperparameters, the kernel, the weight for exploration and exploitation. How much tuning did you have to do, and does the tuning actually matter in terms of the efficiency of the sampling?
So, the only thing that was adjusted in this during this execution was actually the depth. So the goal of the GP and its hyperparameters was to give as good as accurate as possible map of the depth of the highest concentration of phytoplankton inside of this volume. So the hyperparameters had a lot to say about how far you spread the information inside of that volume. So the decorrelation scales are very important. And as I said, the correlation scale used in the GP was based on previous work done by a German team looking at the correlation scales of patchiness of chlorophyll, and we also did some simple statistical evaluation of the decorrelation scale using measurements we did at that particular location. And as I said, the decorrelation was, so the chlorophyll patchiness present at that location was around 900 to a kilometer. So that was important. In terms of the resolution of the GP, that was not that important. I think the resolution of the stochastic process was around, I think we discretized the working area into, I cannot remember, I think it was like 30, we had like 30 by 30 by 30 grid. Yeah. So yeah.
So Alex had a question on if you could talk a little bit more broadly about how you think sampling technology can change with AUV hardware in the future. For example, if AUV hardware increases in its speed or range or processing capabilities or miniaturization, how do you think sampling approaches might adapt to the future?
Interesting question. Well, as I said, some of the work, especially around, as I said, we have certain camera technologies that use microscopes. For example, they have neural nets, artificial neural nets running on board, and some of them can be very big. For example, if you upgrade to a more recent NVIDIA computer, you might be able to more accurately count and recognize different types of zooplankton, for example. And it's a very difficult challenge because the pose of these microorganisms can be very difficult. So if they are so small and they are so the features of them are, they're not like a cat or a dog or a car. Some of them are very similar, and some of them appear in a chain or by themselves. So it's the same species, but they might be chained. They might be covered with pollution. So I think one thing that can change with more, with at least more computational and better neural nets is to be able to actually count and recognize more zooplankton species correctly.
So on the related note, Thorn had a question on how did you actually verify the efficiency of your sampling method? You need to, for you to know that, you need to sample every at a very fine-grained resolution to know how well it's working, right? So how did you get the ground truth data to get your efficiency?
So as you, it's not really possible to ground truth it that much, because in order to have a ground truth, you essentially need to visit all the locations inside of the volume and compare it to your estimate. What we could look at was the change of our estimate over time and see if our estimate over time was changing a lot. The currents might have influenced the currents might have been too strong, so that when we are trying to recreate that information, it's really not giving us a realistic map. It's actually making things worse. So the way we sort of evaluated the performance of this was essentially to look at how much our estimate changed over time, and we saw that the estimate of the internal volume and the internal distribution was fairly constant over time, and it followed it followed what we believed to it was as we expected. We did, of course, computer simulations where we could control the amount. We could sort of simulate this in the computer, where you knew the location, we knew the value at all the locations, and we could test and recreate this in simulation. But when you go out to the field, there is always new things that can surprise you. So comparing this to the performance we have seen in simulation and seeing that things were fairly constant over time, we were certain that it at least gave not a very wrongful impression.
So another question which Ian had, which I think would be more, I can make it a little bit broader, is it seems like the system can also be used for mapping oil spills. So the question which we had is like, how do people map oil spills today? Do they use something similar to your technique, or what else do they use? And other question that Ian has is, at a high level, what kind of a software changes do you have to make to go adapt your technology to any new technology, any new application like oil spills? What do you think is the path for that?
So, of course, you could simply hot swap the oil droplet concentration for the chlorophyll A concentration and try to make a volumetric map of how the oil spill is evolving. And I think, I'm not sure about this, but I think they use AUVs to map oil spills already today. And in those cases, they have several AUVs. They even they have very good estimates of the currents. They have ocean models predicting the current distribution and the dispersant of oil, and then they ground and then sort of verify this with point measurements from AUVs. And in that case, this could be used, but they somewhat brute force it by having enough assets to sort of fully cover the area of interest. And so I think in that case for oil spill, what could be interesting is oil spill response. So on the short term, try to see how an oil spill is evolving. And I have another paper together with a colleague where we are looking at mine trailings. And there we use an ocean model and a more temporal scheme to see how that an oil spill or a mine trailing evolves through time, where you have limited amounts of resources. So that if you go on my Scholar profile, I think there's a paper about that there, but it's about mine trailings.
I see. There's a question from Ethan about did you encounter any issues about changes in the environment over time while you were running the experiment? For example, currents changing between the exploration and the exploitation phase, and did you have to adapt for it?
Yeah, it's a relevant question. So these missions took around 1 hour. So the current situation did not change in that short time. It can change, but in a situation where it changes that fast, you're in a very dynamic and very special oceanographic situation, I think. If you're doing longer missions, that might become a problem, but for doing longer missions and larger areas, I think one has to reconsider using maybe more AUVs, or even consider using more sophisticated statistical models that take into account things as tidal currents and maybe even ocean model data at some level.
See, this is extremely informative. Thanks a lot for taking the time. I think we have covered almost all the questions on the chat. But thank you so much for taking the time. I think it's pretty late for you right now, and where you are.
It's a bit late, but I'm just glad someone is interested in the work.
Yeah, it's a very promising piece of work, and it clearly is. I mean, oceans are huge. We kind of forget how big oceans are, and understanding them requires, you can't really go deploy millions of sensors in the oceans because that's going to be electronic waste. So what kind of work you're doing seems to be extremely important in smart sampling of the ocean so that we can learn about various aspects of what's happening over there. So great work, and thank you so much for taking the time.